Hello. So far, we have talked about genes, gene alleles, their segregation, and how are the phenotypes formed. Today, we will talk about genetic theory and how inheritance underlies. Today, we will briefly talk about chromosomes and the chromosome theory of inheritance. Initial breeding experiments and advances in microscope technique provided evidence for the chromosome theory of inheritance. Proper development requires accurate transmission of genes and accurate maintenance of chromosome numbers. We already have discussed cases of disorders that are caused by either transmission defects or defects in number of chromosomes. Before chromosome theory, there was very abstract understanding of the genes and the physical reality of it. There are at least two noteworthy experiments that worth to mention. One is conducted by Anton van Leeuwenhoek, which by developing microscopy technique was conducted when they looked at semen and they realized that it contains cells, spermatozoa, and it gave them ability to hypothesize that sperm may enter egg, which will cause achievement of fertilization. Next leap forward was the observation of fertilization in frogs and in surgeon, where the coming together of nuclei of eggs and sperm was observed. And the conclusion was made that something in nucleus must be containing hereditary material. Improved microscopy technique allowed scientists to visualize the nucleus and actually to see the movement of bodies through the cell division, the chromosome thread movement through cell division. And so here we were dealing with mitosis, where we are seeing how chromosomes are duplicating within one cell, and then that cell is being divided into two, and it's causing a diploid cells, and there is meiosis where we have duplication of chromosomes within one cell, but then that cells is dividing into four offspring cells, each one of them containing only the half number of chromosomes, and those are the haploid cells. Most cells are diploid. However, when there is a meiosis process, it is causing haploid cells, which means that the cells are containing only the half of the amount of chromosomes. In human, for example, in general, we have 46 chromosomes, but sex cells are haploid cells containing only 23 chromosomes. Fertilized eggs carry matching sets of chromosomes. They have complete set coming half from maternal and half from paternal gametes. Gametes are haploids. They carry single set of chromosomes. They are haploids. Zygotes are diploids. They are carrying two matching set of chromosomes. Mitosis ensures that all dividing cells are developing individuals that have identical full set of chromosomes. Chromosomes can be metacentric or acrocentric. Metacentric are those chromosomes that have centromere where two sister chromatids meet in their geometric center. And the acrocentric chromosomes are those in which centromere is not in their symmetric geometric center. In here, these two chromatids are sister chromatids, where this chromatid and this chromatid are non-sister chromatids. Metacentric and acrocentric chromosomes are non-homologous chromosomes, where two acrocentric or two metacentric are homologous chromosomes to each other. Homologs contain the same set of genes, but they can have different alleles for some genes. Non-homologs carry completely unrelated sets of genes. Karyotype micrograph of stained chromosomes arranged in homologous pairs. Sex chromosomes, unpaired X and Y chromosomes. Autosomes are all chromosomes except for X and Y sex chromosomes. Cells of each species have a characteristic diploid number of chromosomes. Here is a map of a human karyotype. As you can see, we have a full set of chromosomes, 22 pairs of regular chromosomes and 1 pair of sex chromosomes. Here we can show that this is chromosome 21, where in Down syndrome, about which we have talked previously, the person would have one extra pair of 21 chromosome. So here is a question. What do you think? This human karyotype, is it male or female? So Sutton studied meiosis in great lubber grasshoppers. So before meiosis, testis cells had 24 chromosomes, 22 in matched pairs, autosomes, and 2 unmatched, which were X and smaller Y chromosome. So after meiosis, 2 types of sperm were formed. By 50-50 chance, there were 11 chromosomes and an X chromosome, and 11 chromosomes and another 50% chance of Y chromosome. In case of X, there were all 11 chromosomes plus an X chromosome. So, which means one chromosome pair determines sex. So, fertilized egg with sperm carrying an X chromosome will cause in XX female. Fertilization of egg with sperm carrying Y chromosome will cause in XY, which is male. Sutton concluded that X and Y chromosomes determine sex. Children receive an X chromosome maternal from mother and either X or Y chromosome from their father, which is going to determine the sex of the child. Results are always in 1-to-1 ratio of females to males. So let's look at the difference of sex determination in fruit flies and humans. In Drosophila, ratio of X chromosomes to autosomes determine gender. In humans, presence or absence of Y chromosomes determines gender. Abnormal numbers of X or Y chromosomes have different effects in humans and flies. Here is the complete table. For Drosophiles, XXX will cause in death. XX will be normal female. XXY will be also normal female, where XO will be a sterile male, and XY will be normal male. XYY again will be normal male, and OY will cause in death. For humans, XXX nearly normal female. XX is the normal female. XXY is Klinefelter, which is the sterile male. XO is Turner female, which is the sterile female. XY will be normal male. XYY will be normal or nearly normal male, and OY will cause in death. So now let's look at the different mechanisms of sex determination for different species. So we know heterogametic sex is gender with two different kinds of gametes, XY males in humans, ZW females in birds. Homogametic sex is where gender with one type of gamete is determined. For example, XX females in humans and ZZ males in birds. In some species, gender is determined by environment. For example, temperature. Let's look at this table. So in case of humans and Drosophila, for XX we will have female, and for XY we will have male. Moths and C. elegans, in case of XX we will have female, and XO we will have male. Birds and butterflies, ZW will cause in female and ZZ will cause in male. Bees and wasps, diploid is the case for female and haploid is the case for male. Lizards and alligators, cool temperature will cause in females and warm temperature will cause in males. Tortoises and turtles, warm temperature is for females and cool temperature is for males. Anemone fish, older adults will have females and young adults will have males. So during the cell division, there are gap1, G1 phase, synthesis S phase, and gap2, G2 phase, which seem to be that cells are doing nothing, but they are actually very active during that process, and that the actual cell division process is happening with duplication of the DNA and then polarization of DNAs into two different poles, two opposite poles, and the actual cell division and formation of two duplicate cells. So here is the diagram for the cell division. So there is the interface, as we mentioned, with G1, S, and G2, and then there is the actual cell division process, which is the mitosis and cytokinesis. The cytokinesis is the actual cell dividing part phase. So let's summarize what we have talked. So G1 phase is where the chromosomes are not duplicating or dividing. Length of time varies in different cell types. Then there is S phase, duplication of chromosomes into sister chromatids. And then there is G2 phase, where synthesis of proteins required for mitosis. So mitosis has 5 different stages. Let's go over those 5 stages. So there is stage 1, prophase, where chromosomes condense and become visible. Then there is stage 4, prometaphase, where spindle forms and sister chromatids attach to microtubules from opposite centrosomes. Then there is the metaphase, where chromosomes align at the cell's equator. And then there is the anaphase, where sister chromatids separate and move to opposite poles. And finally, there is the telophase, in which chromosomes decondense and are enclosed in two nuclei. So here is a nice visualization of the prophase. Here is a very nice visualization of the prometaphase. On the left picture, the red is the environment, and then the yellow are the fibers, and blue is the DNA. Here is a very nice visualization of the metaphase, and as one can see, we are observing centralization of the DNA on the equator of the cell nucleus, and then also we can observe the coming of the fibers towards the DNA, the chromosomes, where they are going to get attached to the centromeres in order to pull them towards the poles, opposite poles. And here is the anaphase stage of the mitosis. In both pictures, we can see how fibers are pulling two sets of chromosomes from the center of the cell towards the opposite poles. And this is the visualization of the telophase. In two different pictures, we can see the formation, the beginning of the formation of two separate cells. We already can notice the two nuclei and the spindle fibers disperse. The cytokinesis is the final stage of the cell division. It begins during the anaphase, but it's not completely over until after telophase. So the parental cells split into two daughter cells with identical nuclei. Here is a comparison of the cytokinesis for plants and for animals. So in case of animals, there is a formation of cleavage furrow during the duplication or cell division, and in plants case, instead of cleavage furrow, there is a frame formation around the cell, which is basically made out of sugar. In some organisms, cytokinesis does not always occur after mitosis. This is a good slide that identifies what we just have talked about. Please go very carefully through this slide and understand what was clear for you, what was remaining unclear, so that you can go back and go over the material in order to understand everything.